Dear student, due to coronavirus, school started online classes for your study. So you can subscribe Golden Flower Palia YouTube channel, like and press the bell icon.
Hello students, after the completion of the formation of primary endosperm nucleus and the formation of zygote. So next topic for you, this is the embryo formation in angiosperms and it is also known as embryogeny. So, in simple words, we can write here the development of embryo from diploid zygote is called embryogeny. So now student, question arises here, how embryo is formed? So dear student, suppose that this is a one diploid zygote. Now this zygote divides mitotically. So it forms two cells, upper and lower. Upper cell suspensor and lower cell embryonal cell. Both the cell again divide mitotically. It is a transverse division and it forms upper cell transverse division while lower cell divide longitudinally like that. So this is a four-celled embryo. Four-celled embryo. Now upper cell again divide transversely and lower cell again divide transversely. So in next phase, you got here upper four cell like that and lower or globular means first longitudinal division and then it is a transverse division. To total here 8 cell embryo having 4 upper cell and 4 lower cell. So this is a zygote, B 2 cell, C 4 cell and D this is a 8 celled embryo. Now upper cell again divide transversely but lower cell divide periclinally. So it form 8 cell globular embryo. So next here, like that. Upper cell, then remaining cells are there. 1, 2, 3, 4, 5, 6, 7 and 8. And downward a globular 8-cell embryo form, like that. So here, student, this upper cell is known as posterior cell. This is the suspensor, and its lower cell, this is a hypophysis. Now this is a globular, also known as octant stage. Octant stage. So this is the structure E. Or now this is the F. So in this phase, upper this is the haustorium, then long suspension, then upper this is the radical, then last this is the dermatogen, this is the procambium. After the more and more development of dicot embryo, it forms a heart-shaped structure. So this heart-shaped structure having two cotyledons. So you have seen these are cotyledons. This is the haustorium. This is the suspensor, and this is the radical, and last this is the end of plumule. So this is the structure G, embryo development in dicot plants. So student, about embryogeny or embryo development, so we have taken here dicot plant having two cotyledon. Dear student, the development of embryo from zygote, though having diploid structure. So this is known as embryo development, embryo formation or embryogeny. One thing more clear here, the development of embryo here, meroblastic, a word came here, meroblastic means embryo developed from zygote. Second, endoscopic. It means embryo developed interior of the suspensor. So after that a pro-embryo is formed. In very brief, zygote divide mitotically to form two cell structure, suspensor cell and embryonal cell. Further divide transverse division and longitudinal division. This is a 4-cell embryo. Again transverse and again transverse. So this is a 8-cell embryo. Now further proceeding like that. One thing more here, the formation of hostorium or hostorial cell. The main function of real cell is to take nourishment from the endosperm and provide it to the growing embryo. Dear student, when cell is the egg cell, this is the embryonal mass having two tier, upper one epivessel and lower one hypovessel. Epivessel form radicle and plumule, while hypovessel form dermatogen and procambium. Or next, when fully mature embryo is formed in a dicot plant. So this fully mature dicot embryo has one hostorium for the nourishment of the growing embryo at the micropylar end. One long suspensor. This is a connection between endosperm to embryo. Then having two cotyledons along with radicle having one root cap, plumule having a covering sheath. So dear student, in future radicle develops into root, means the development of root or formation of root, and the development of shoot or stem, it is from plumule, means to say this plumule in future develop shoot and this radicle form roots. So this is the future of embryonic cell stage. Octant stage. Again divide periclinally to form outer dermatogen, it is also known as protoderm, and inner cell again divide to form plerome, procambium. So procambium further divides to form vascular tissue or steel, while plerome forms plumule, and last is, though dermatogen is there, it forms a protective layer known as epidermis. And the cells of ground tissue further divides to form cortex and pith. So this is about future of embryo. Dear students, in embryo development only mitotic cell division carried out. So this is for you. Thanks.
Do self-study at home and stay home, save lives. Thank you very much.